Let's continue with our lecture on the kinetics of biological processes. We have deducted the Monod equation from enzymatic reactions and described the reaction inhibitions by modifying the Monod equations. The growth kinetics of certain microbial communities is also largely affected by pH, such as AOB, NOB, and methanogens. pH in the environment influences proton balance, therefore ATP production, which is driven by proton concentration gradient. pH also affects the ratio of protonated and dissociated compounds, like ammonia and acetic acid. Their protonated form can be substrates or inhibitors, or both, for certain groups of microorganisms. For example, free ammonia is both the substrate and inhibitor for AOB during nitrification. High concentration of free ammonia also inhibits methanogens during methane production. These protonated compounds can diffuse through cell membrane and dissociate in the cytoplasm that has a different pH than outside of the cell to release proton and disrupt the proton balance. This figure shows the effect of pH on the growth rate of AOB. It can be described by this empirical equation. Temperature is also known to affect the rate of chemical reactions, including biochemical reactions, based on thermodynamic principles. Recall the free energy change at the standard condition for the half reactions delta G0. The free energy change depends on the temperatures. The effect of temperature on the reaction rate can be described by Arrhenius' equation. If we take the ratio of two reaction rates at two temperatures, we can use this equation to calculate the reaction rate at any temperature if we know the reaction rate at a standard temperature. Because we're not looking at extreme temperatures for biological reactions, R times T1 times T2 can be considered as a constant that we can call natural log theta. Then the equation can be written as this. The active cell biomass will eventually decay. There are two major decay models that we will discuss in more details. The traditional decay model, where the active cells decay to become cell debris XD. There's transfer of energy just like during the growth process, hence loss of COD in this process. The other model is the lysis regrowth model. The active biomass lies to form debris XD and particulate substrate XS without transfer of energy or loss of COD. The particulate substrate degrades to form soluble substrate, which is utilized for cell's growth. This is a reaction describing the traditional model. Biomass reacts with electron acceptor, which can be oxygen, to form CO2, reduced acceptor, which can be water if oxygen is the electron acceptor, nutrient, and biomass debris. We can write the stoichiometric balance of this reaction in COD. FD is the fraction of active biomass electron that goes to debris. You can recall the similarity of this to the biomass growth model. The decay rate of biomass is the first order of biomass concentration. B is the specific decay rate. Its unit is per time. Then we can write the kinetic equation for debris production and oxygen utilization based on stoichiometry. This is a reaction of ammonia release from organic nitrogen during decay in the traditional model. Biomass nitrogen becomes ammonia nitrogen and debris nitrogen. We can again write this in COD terms. INXB and INXD are the nitrogen fraction of COD in active biomass COD and debris COD, respectively. Because 1 unit of biomass COD contains INXB unit of nitrogen, and 1 unit of biomass is converted to FD unit of biomass debris, which contains INXD times FD unit of nitrogen. So a mass balance tells us 1 unit of biomass would release INXB minus INXD times FD unit of ammonia nitrogen. We can get the release rate of ammonia nitrogen according to stoichiometry. In the lysis regrowth model, because there's no loss of COD during lysis, the lysis reaction can be written as this. 1 unit of biomass COD is broken down to form 1 minus FD prime unit of particulate substrate COD and FD prime unit of biomass debris COD. FD prime is the fraction of active biomass that becomes debris. Again, we know the decay rate is first order of biomass concentration. BL is a specific lysis rate with a unit of per time. Then we can write the debris production rate and oxygen utilization rate according to stoichiometry. As for the nitrogen in biomass, it becomes particulate biodegradable nitrogen and debris nitrogen. The stoichiometry of releasing particulate biodegradable nitrogen in the lysis regrowth model is similar to that of releasing ammonia nitrogen in the traditional decay model. We can get the kinetics equation for the release of particulate biodegradable nitrogen according to the stoichiometry. The stoichiometry and kinetic expression for the lysis growth model appear to be similar to the traditional decay model. But the particulate substrate produced will be hydrolyzed to become substrate, which will be utilized for energy generation and cell synthesis in the growth model. Therefore, BL is larger than B, and FD prime is lower than FD. In the lysis regrowth model, the particulate substrate generated after cell lysis, along with other particulate substrates in the feed, cannot be directly utilized by the biomass, but will be broken down to soluble substrate through hydrolysis by extracellular enzymes. There is no COD loss in the hydrolysis process, as it is not a redox reaction. The kinetic description of hydrolysis is not well understood. A lot of models just use a first-order reaction to approximate it. Another expression can be Monod-type reaction. Instead of substrate concentration, the reaction rate depends on the ratio of particulate substrate concentration XS to the active biomass concentration XBH. Kh is the maximum hydrolysis rate, and K sub x is the half saturation coefficient. The generation rate of soluble organic nitrogen is proportional to solubilization rate. XNS is the particulate biodegradable nitrogen. Another process we're talking about is the formation of soluble microbial products, including the extracellular polymeric substances, EPS. Because it plays a vital role in the formation of sludge flux in suspended growth reactors and in the formation of biofilm in attached growth reactors. The non-growth associated formation of SMP is the result of decay, while the growth associated formation occurs along with cell synthesis from the electron donor. To get the stoichiometry of non-growth associated formation of SMP, let's first recall the stoichiometry of traditional decay model. Then we can write the stoichiometry with SMP formation. FMP is a fraction of biomass electron goes to form SMP. The kinetic equation is based on the stoichiometry. the non-growth associated formation rate of SMP equals to the specific decay rate times FMP times active biomass concentration. For the growth associated formation of SMP, let's recall the stoichiometry of cell growth. We can include the formation of SMP in this equation. YMP is the yield of SMP with a unit of COD of SMP formed per COD of substrate consumed. We can transform this by dividing both sides with the true yield YH. According to the stoichiometric relationship, we can write the kinetics equation for the growth associated formation rate of SMP. We previously only looked at the true yield, which considers that all energy is used for growth. The observed yield is the actual increase of biomass COD per unit of COD consumed, considering cell maintenance and decay. We can get the observed yield from the true yield and specific decay rate. So the observed biomass growth rate is a result of true growth and decay. The true growth is the true yield times substrate consumption rate. Decay is the first order biomass concentration. The observed yield is the observed growth rate divided by the substrate consumption rate. Substitute the observed growth rate can get this. When true growth rate equals the decay rate, there is no change of biomass concentration. All energy generated from energy reaction is used for maintenance. The substrate utilization per unit of biomass is called the maintenance energy coefficient, M. It is also the specific decay rate divides the true yield. When substrate utilization rate per unit of biomass is smaller than the maintenance energy coefficient, which is B over Y, the observed yield is less than 0. Then the biomass is starved as there is not enough energy to sustain the energy needs of the biomass.